My name is Natalia Elizalde, CBO at Vive Biotech. So, at Vive Biotech, we are a CDMO specialized on lentiviral vector, doing development and manufacture of lentiviral vectors from scratch up to potentially commercial stages. Yeah, for sure. Well, on this advanced therapies event, there is a strong focus on viral vectors and manufacturing that, from my point of view, is a very positive sign because it reinforces the fact that viral vectors continues being the preferred option for gene transfer itself, and also because it's recognized how important manufacturing is in order to contribute to the success of the therapy itself later on to be approved for commercial stages. Yeah, well, I tried to attend to several kind of panel discussions and so on, especially the one focused on cell therapy. They were very interesting because, as you know, lentiviral vectors, they are being used mainly for cell therapy modified manufacturing. Also, I attended to some presentation focused on the in vivo use of lentiviral vectors, which is so positive because some years ago, like 2-3 years ago, this was something very unique. And now it's being more and more frequent. Indeed, for instance, for us as a company, we are already working on 8 programs using lentiviral vector for in vivo. And these are the 2 that I would like to reinforce or to really highlight. But also there was one specific talk on the use of these kind of vectors for immunotherapy. And this is something also that is being strengthened in the last years, and that we are seeing within the application of the viral vectors for these kind of applications. Yeah, the next generation vectors, from my point of view, how they will look like is using different kind of pseudotypes, a part of the classical VSVG. This is something that we are really also observing as a CMO focused on lentiviral vectors. So pseudotypes that are really targeting the cell of interest. Moreover, there are different kind of generations of vectors that are being more and more popular, such as the 4th generation ones, using 4 packaging plasmids instead of 3, or for instance, those vectors using packaging cell lines or modified cell lines for its production. So from my point of view, this would be the trend, and this would be how the next generation vectors will look like. There are different ones. I mean, this was a very useful conference itself. I had the chance to meet with several of our clients, so for sure this is something very important, but even more, there were that many panels focused on different interests. For instance, for me, the one for viral manufacturing was so interesting, the one for cell therapy as well. So I tried to, respond during this interview on the topics that were of higher interest for us as a CDMO, but ensure that this is an event interest not only for companies as ourselves, but also for therapy developers or other kind of service providers.